are 4 macromolecules, okay? Because we need to be able to differentiate between them. Your job as an AP Bio student, one of them is to know what a macromolecule is and how you tell them apart, okay? So first of all, macromolecules. There are questions, and they'll say, which macromolecule, and then they'll have a question with it. But if you don't even know what a macromolecule is, you already have a problem. Okay, so which macromolecule, and then if it says that, you know already that you only have 4 options. There are 4 macromolecules in biology that we discuss. Lipids is one of them. Okay, so lipids is the one we have here. What else do we have? What is another macromolecule? Lipids and what is the primary structure of a protein?
Just the base pairs, right?
Not base pairs, but amino acid order.
Oh,
just the order of the amino acid, which I guess is from the base pairs in DNA. But, right, the order of the amino acids, primary structure of protein, right? Secondary structure of protein. Anybody remember anything about secondary structure of protein? Yeah.
Isn't it kind of like a spindle?
It can be an alpha helix, yeah, or it can be a beta pleated sheet. It could be one of those two. Does anyone remember what kind of bonds there are for that? Is that in between the R groups or is it in between the side chains? Here is the process, and I don't know if you remember this from before, but we're going to stick it in our brains good and solid right now. It's called dehydration synthesis. Does that ring any bells with anyone? Dehydration synthesis. So what does synthesis mean? What does synthesize? If you synthesize something, what are you doing?
You're making something.
Making something, yeah. So synthesis is just making something. So we're making something, and what does it mean to be dehydrated?
Take the water out of it.
Yeah, we're gonna take water out of it. All right, cool. So what we're gonna do is we're gonna take our glycerol, which has little hydrogens all over it, and we're gonna take one of our triglycerides. We're gonna do this 3 times. First of all, you gotta figure out, you want the little tails to go down from them, and so you gotta figure out which one of your lipids goes where, or your fatty acid chains goes where. So basically what happens is when 2 molecules get close together here, there's a charge. So this little guy, hydrogen, is positive right here, and this little guy, hydroxide, will be negative. So they have a charge, and they like each other. So pop, they come off and reform. I'm trying to do this with not enough hands. Reform into water, yeah? And then meanwhile, there's an open binding site there, so they can be bonded together. And voila! We have begun the process of making our fatty acid chain into an actual lipid. Okay? So then you grab another one, and you're like, all right, where does this one go? Looks like it'll go right there and go straight down. That'll be good. Pop off the hydroxide and the hydrogen. Make your lipid. So dehydration, there it goes, yeah? And synthesis. Yeah, dehydration synthesis. Okay, so they all line up, yeah? So they would go line up, and if we wanted to spread them out on the floor, we could make a big one, but COVID. Speaking of which, I'm going to cough, just so you can have a visual. So lots of them this way, right? Lots of them this way, 3D, lots of them this way, whole sheets of them. Also, it's a bilayer, so, right? Okay, here's your attractants. First of all, the lipids are slightly attracted to each other. Okay, so the lipids like each other. That's good, all right? Here's your repulsions that make it stay this way. The lipids hate water. They are protected from water in here. However, if you made it where all of a sudden you just had air on this side, instead of water, they would still, like down here, they would still form this way because also the lipids are repulsed from the phosphates. Okay, so they're never going to have the phosphate right with the lipid. They're going to have it lipids together, phosphates on the outside. Okay, so that's super important because that's what makes it always remain in a bilayer. So 4 kinds of macromolecules. Let's review really quick. Give me one.
Lipids.
Lipids,
Protein.
proteins,
Nucleic
Nucleic acids and carbohydrates. How can I tell the difference between a carbohydrate and a lipid? Okay, did you learn something today?
Yes.
Okay, that's good. No, I gave up on that. All right, put everything back in your bag and go wash your hands. You have 4 minutes to complete that. If you think about it, it would be like if the repressor was there, it would just like sit there on the operator. And if the RNA polymerase binds to the promoter, well, it can't move. It can't move downstream because there's something blocking it. That's it. That's basically it. It's not any sort of like really challenging chemistry to get there. It's just, excuse me, you're in the way. I can't move. That's how the repressor works. Okay. So that's pretty straightforward. So those are the parts of the operon. You need to know them. You need to know how it works. And then you need to be able to read some nice diagrams